Hi everyone, welcome to Chapter 5. Chapter 5 is where we're going to start to get into many of the different types of eukaryotic microorganisms that we will discuss later on in the chapters throughout the semester. Chapter 5 looks at our fungi, our protozoan, our algae, and our helminths, and steps away from the bacteria and archaea for a little bit to take a look at some of the main characteristics of those organisms. Obviously, this chapter is not going to be a comprehensive overview as it's more of a survey chapter to allow us to be able to understand some of the key characteristics that comprise those different groups. So I hope this finds everybody doing well, and here we go. So one of the big concepts that we need to understand is how did modern eukaryotic cells evolve? And this occurred through a particular theory which we refer to as the endosymbiotic theory. And what the endosymbiotic theory involves is that at one point you have this larger prokaryotic cell back millions of years ago. And at some point, it actually engulfed a smaller prokaryotic cell. And when it engulfed the small prokaryotic cell, this smaller prokaryotic cell actually started to live inside of this larger cell. And we now know today, based on studying different evolutionary trends as well as some of the different properties, we now know that eukaryotes acquired their mitochondria from a rickettsial bacterial ancestor, so a member of the Rickettsia genus. And the chloroplasts that we find inside of a eukaryotic cell were actually acquired from cyanobacteria. So these little photosynthetic bacteria, they have this green pigment to them, were engulfed into this larger prokaryotic ancestral cell and eventually became the chloroplasts that we know of today. And there's a lot of different characteristics that support this theory, such as some of these organelles having their own DNA nucleic acid source, which is different from any of the other organelles that we find within a eukaryotic cell. So as we mentioned, we're going to talk about really 4 different groups today. We're going to talk about the protozoa, which are usually unicellular. We will get in and look at the fungi and the algae, which can be either unicellular or multicellular. And then we get into the most complex group of all of the microbes that we will study, and that is the helminths or the parasitic worms. And they are all multicellular, some of them even having primitive organ systems that we would think of in a typical mammalian organism, things like digestive systems and some very simple nervous system-like structures. So here's our eukaryotic cell, much, much more complex than what we see with the prokaryotes. We have all of these different mechanisms inside the cell that comprise the organelles. And if you think back to chapter 1, remember that the whole point of and the difference between a eukaryote and a prokaryote was the presence of this nucleus, this membrane-bound nucleus, as well as all of these different organelles that execute various functions within the eukaryotic cell. So a much more complex nature to the eukaryotes. So we're going to get in and we're going to talk about several different external structures, going back to ones that you're already familiar with, that extra slime coating on the outside of the cell, the glycocalyx, which can either be a capsule, that nice, smooth, defined structure, or the loosely bound slime layer. And we'll also talk about some of the boundaries of the cell, things like the different components that make up the cell walls of these different organisms, and the structure of the cytoplasmic membrane, this phospholipid bilayer that regulates the movement of things in and out of the cell, key for transport. We'll also get in and start talking about the general function. I'm not going to spend much time on the organelle functions because you have had these in previous courses. but we will look at some of the different eukaryotic organelles that drive functionality within the eukaryotic cell. We'll also talk about some of the locomotion in terms of things like flagella and cilia. We'll also get in and talk about, when we get into the protists, we'll talk about this primitive foot-like structure called the pseudopod, which gives certain protists this snail-like movement to them. So let's start off first with the external structures, and specifically with locomotion or movement. So similar to bacteria, some eukaryotic cells also possess this whip-like tail known as the flagella. And the flagella are a little bit different in eukaryotes in the fact that they have microtubules, these kind of protein-like structures that extend through the body of the flagella. And they are actually covered by the cell membrane, kind of extends from the cell and covers the flagella. Cilia, on the other hand, are these tiny little bristle-like or hair-like projections that beat back and forth. So they actually move to the left and then to the right. So they do this alternating side-to-side movement, which is known as a stroke. And they are similar to the structure of flagella, but they're much more shorter and they're more numerous across the surface of the cell. And really their job is to support things like feeding and motility of some of these microorganisms. So you'll notice that this particular protist here actually has an oral groove, which serves kind of as the mouth. And the little cilia actually help to channel the food into this oral groove to supply the microbe with nutrients. We also talk about the slime or glycocalyx layer, this protective boundary on the outside of the cell. And the key thing to really understand is its composition is mostly polysaccharides, these sugar-like materials. And we mentioned that they can either be loosely associated in a slime layer or very firmly organized, as in the capsule. And their job is to really help support adhering to surfaces, protection from phagocytosis, which is that engulfing by the white blood cells, and also has some role in signal reception. The fungi and the algae have some of the thickest cell walls, and some of the protozoa and algae lack cell walls and only have that membrane layer. So we're going to really break that down, that these fungi and algae can have a very rigid, thick cell wall, but things like protozoa, the protists, and the algae lack that cell wall and have just a cell membrane. So let's talk about the cell wall. Some of the key things that I want you to walk away with on the slide is, again, it is responsible for providing that support and shape. Fungi are composed mainly of chitin, which is this very thick material. It's the same kind of material you would find in the shells of crustaceans like lobsters or crabs. Or sometimes cellulose, which is a component that is added to paper. Algae, on the other hand, actually include the cellulose, but have some other different components that comprise their cell wall. The cell membranes can also differ a little bit as well. Typically, the cell membrane or the cytoplasmic membrane are those phospholipids. And those are those little head groups with the tails that come off. The head groups are hydrophilic, meaning that they're attracted to water. The tails are hydrophobic, meaning that they are water-faring. Sometimes things like the fungi can have these components called sterols, which actually help to stabilize and support the structure of the membrane. So here's some of the different organelles that we're going to go through quickly. Again, know their overall function, but you don't have to get too deep into structure and things like that. So lysosomes are responsible for containing digestive enzymes that help to digest food particles and protect against invading microbes. Vacuoles are these little membrane sacs that are responsible for helping to store in materials. Phagosomes, again, will merge with the lysosome. And when we talk about the phagosome-lysosome structure, these are usually designed to destroy foreign substances to the cell. The cytoskeleton is, again, we talked about the microtubules. It's this series of filamentous type materials that are involved in not only movement, but also structural support and transport of things within the cell. So it has a really important function to it in terms of being able to not only support the structure, but also supporting movement of materials within the cell. Again, I'm not going to go crazy on this. You don't really need to get into comparisons for prokaryotes and eukaryotes. Just be familiar with some of the key differences which we've talked about between Chapter 4 and now Chapter 5. So we're going to get in and start taking a look at some of the eukaryotic microorganisms. So we're going to start with the fungi. And as we mentioned, the majority of these are unicellular. However, there is the ability for some to be multicellular. And there is a ton of diversity in this particular group. Over 100,000 species that are arranged into two different groups. The macroscopic, or ones that we can see with the naked eye, things like mushrooms and puffballs, and the microscopic fungi, which we're going to focus on in this course, that include things like the molds and the yeast. And the molds are arranged in these long, filamentous, almost like hair-like structures that we call hyphae. And we're going to see that when we take a bunch of these long strands and we put them together, many hyphae produce a mold mat called a mycelium. The yeast, on the other hand, are these unicellular, kind of oval-shaped fungi that go through asexual reproduction. What that means is there's no merging of a sperm and an egg. The asexual reproduction in a yeast is a process called budding, where a little piece of the yeast cell actually breaks off and creates a new cell. Very importantly, we're going to see that some of the fungi are what we call dimorphic, meaning that they can actually alternate the form that they exhibit, either the yeast or the mold form, depending on things like temperature. So we mentioned the hyphae are these kind of long, thread-like structures that we see. And when we take this whole group of these thread-like structures, we form this little mold mat here, which we refer to as a mycelia. And the mycelia is this combination of all of these different hyphae grouped together. We mentioned the yeasts have this kind of oval-shaped appearance to them, and they reproduce asexually through this process called budding, where a part of the outside surface of the cell actually grows and breaks off to form a new yeast cell. Fungi are heterotrophs, meaning that they have to acquire their nutrients from organic matter. So the majority of fungi are saprobes. And if you remember, when we talk about a saprobe, that means that they can feed off of dead, decaying organic matter. They're decomposers. Bacteria and fungi are the two big members of the microbial family that serve as these saprobes. Like most microbes, the majority are harmless. However, some of the fungi are parasites. And we call these different parasitic infections mycoses. So things like athlete's foot is an example of a mycosis. It's a fungal infection of the body. And we can see things like superficial mycoses, like athlete's foot. And we'll also talk about much more systemic and deep-seated mycoses, infections that can actually become systemic and spread throughout the body. Fungi reproduce through spores. And we're going to talk about asexual and sexual spores tonight. So we'll start with the asexual spores first. And there's really two members of this group. We have what are known as the sporangiospores, and that's because they grow in this, almost looks like a lollipop, this kind of sac-like structure that contains all the spores, known as the sporangium. And when that sporangium bursts or lyses, it is going to release all those spores out into the environment. And spores are so incredibly light that they can actually be carried through the air by the wind from one place to another. The conidiospores, on the other hand, are free spores. So really what this looks like is, if you think back to our hyphae, basically in a conidiospore, you're going to get little pieces of the ends or the tips of this hyphae that are going to break off and serve as the spores being carried from one place to another. So we have our sporangiospores, which are in that sac-like sporangium, and the conidiospores, which are free spores. So here's the examples here. Here's our sporangiospore, this kind of lollipop-like structure that can lyse and break open and release all of these little spores. And then we also have the conidiospores, which here's our hyphae. And you'll notice that the ends of the hyphae, the tips of the hyphae, actually fragment off and get carried from one location to another. So sporangiospores are contained within a sac. Conidiospores are not. They are free-formed spores. Sexual spores, on the other hand, are consisting of several different groups. And the three groups that I want you to make sure that you know fit into this category are the zygospores, the ascospores, and the basidiospores. And what I want to do is focus really on the ascospores, are one of the most important ones. So our zygospores, again, have that little sporangium-like structure. And when the sporangium lyses, the spores can travel from one place to another. But remember, since these are sexual spores, there is the combination of what they call a positive and a negative mating strains that come together to form the mature spores that eventually break open and get released. The ascospores are a little bit different. These ascomycota are asexual conidia. And what that means is they are asexual in that they break off. They don't require a positive and a negative to come together. However, they are sexual spores that occur within these little pieces of the hyphae. So you'll notice that you've got some of the blue and some of the purple resulting from the combination of these strains. You'll also notice that there's these little divisions throughout the hyphae. Those are what they call septae. And these little septae, or walls, actually divide the hyphae into different regions. And then we have the basidiospores, which are found in things like the mushrooms, and will actually fragment off. And once they're fragmented off, the germination of those mating strains will come together through fertilization, and that will lead to this growth of the mycelium, that mold mat. So again, make sure you're familiar with the different groups, specifically the zygomycota, the ascomycota, and the basidiomycota. We also talk about the fungi imperfecti. And the fungi imperfecti are important because They're classified by whether the fungus lacks a sexual state or not. So how do we identify fungi? Well, we can isolate them on specific laboratory media, but we also do a observation both through the microscope and visually at things like their spore-forming structures, the type of hyphae that they have, whether they're septate or non-septate, meaning they have those walls, the texture of the colonies and the color of the colonies, their physiological characteristics, as well as looking at their genetic material. So we already mentioned that most fungi are harmless. They don't cause any impact on human health. However, some have an adverse impact, meaning the mycoses. And the mycoses can cause a series of different diseases. We have hypersensitivities, which are basically allergic reactions. We can have an intoxication, which means that a toxin is ingested in the food. Or we can have an infection. Things like we saw with the superficial mycoses, like athlete's foot, are mycoses that are considered infections. Molds, as we know, can also be a source of damage to crops and food. But there are many, many, many beneficial impacts. Things like the penicillium mold are the source of antibiotics. They are able to help generate alcohols, organic acids through fermentation. They're able to produce vitamins. They can help decompose and break down things in landfills oftentimes. And they're also used in the process of making foods like cheeses. So table 5.6, you don't necessarily need to memorize at this point. What I want you to do is just be able to take a look at this and see some of the overview of some of the superficial and the systemic fungal infections that we'll talk about this semester. We're also going to talk about the protozoa. And the protozoa are comprised of two different groups. We have the algae and the protozoa. The algae are usually unicellular, and they are able to go through photosynthesis. The other side of that are the protozoa, which are non-photosynthetic, and they have very, very strict environmental conditions that they require. So the protozoa do require a habitat that is moist. If they lack moisture, it's going to cause the cells to dehydrate and die. So protozoa are found in aquatic environments. They have that requirement for moisture. As we mentioned, the algae are photosynthetic, and they contain the chloroplasts, that organelle that contains chlorophyll that permits the process of photosynthesis to occur. As we mentioned, just like the fungi, they have very thick cell walls that contain materials like cellulose, and they may or may not have flagella. It's not a requirement. Sometimes they are not motile. Most, as we said, live in free living bodies of water, and we refer to those as plankton. And the plankton actually provide the food for most aquatic habitats. They're really the bottom of the food chain for many, many food webs. Algae are actually large oxygenic photosynthesizers, meaning that they produce a high amount of oxygen for the atmosphere. However, there is one condition where the algae can actually cause human disease. And those are through these tiny microbes called dinoflagellates, which result in a condition known as red tide. And when these organisms are ingested, they actually impact the nervous system, the central nervous system, through the toxins that they release, and can actually cause a condition known as paralytic shellfish poisoning, where there's a general paralysis of the body. Also, I do want to mention, as we talk about the protozoa and the algae, one of the big pieces that we're going to see is the red seaweed, which fits into this algae category, is actually the source of the agar that we are going to use in our lab this semester. So that's really important to understand, is that our agar is actually an extract from red seaweed in that algae family. The protozoa, on the other hand, are also a diverse group. We saw that the fungi contained over 100,000 different species, while the protozoa contains 65,000. And these lack a cell wall. So they do not have a cell wall. And for the most part, most are harmless, and they free live in that very moist environment that they require. Some do have the ability to be parasites. We'll talk about Plasmodium, which is the protozoa that's responsible for the disease malaria. And all are heterotrophs. Okay, since they don't photosynthesize, they have to acquire their organic material from other sources. They serve a very important role. They engulf other microbes. So they are going around and not only ingesting bacteria, but algae as well. Most protozoa, as we mentioned, are extremely modal. They have a locomotor structure that is designed to help them be able to move from one place to another. And the three big ones that we'll talk about are the flagella, which is that whip-like tail, the cilia, which are those little hair-like projections that oscillate back and forth to the left and to the right, and the pseudopods, which is that primitive foot, which gives them this kind of snail-like movement. So they extend this projection out, they stick to a surface, and they basically pull themselves forward like a snail. And protozoa can actually exist in two different forms. So this is important to understand. In bacteria, we usually talk about the vegetative cell and the endospore. The vegetative cell is the metabolically active form, and the endospore is that dormant resistive structure that's produced in stress conditions. Well, the protozoa do the same thing as well. The trophozoite is that motile, actively feeding stage, and when they're exposed to stress conditions, they can go into this dormant stage known as a cyst. We see that protozoa reproduce asexually, usually through the process of mitosis. And if you remember, mitosis really contains several different stages, and we refer to those stages as IPMAT. So the easiest way to remember it is interphase, or I, is where we get the DNA to condense into the state known as chromatin, this tightly compacted state. Then we go into prophase. Metaphase is where all of the chromosomes line up in the middle of the cell. Anaphase is where the chromosomes are actually pulled apart towards the opposite ends of the cell. And telophase is where the cell now divides down the middle into two new cells. So in that prophase and interphase, we have the preparation and replication of the DNA, followed by the separation of those chromosomes into the two daughter cells, and then finally the actual division of the cytoplasm starts to occur in that telophase. So things like the eukaryotes are going to go through mitosis for their cell division. Whereas we talked about with the prokaryotes, things like the bacteria, they were going through things like binary fission, where the cell just divides down the middle, a much simpler form of mitosis. So how do we identify protozoans? Well, a couple things. The shape and size of the cell, obviously the type of movement structures, the type of locomotor structures. Do they have flagella? Do they have cilia, or do they have a pseudopod? The presence of those cysts, can we see that trophozoite versus cyst form, and then also the number of nuclei. When we culture protozoans, we usually can do it on artificial media, or we can inject them within lab animals and study for further identification. And these specimens most times are found in places such as the feces. We'll see things like Giardia lamblia. Giardia is a flagellated protozoan that is detected through examination of a patient's fecal matter. And then we also have things like Trichomonas vaginalis, which leads to an STI and is actually sampled from the vaginal tract. So here's our different protozoa based on motility. So we have the mastigopherans, which are flagellated. We have our amoebas, which have this pseudopod or this long projection that's used to stick to a surface and pull the cell along. We have the ciliopherans, which have the little cilia surrounding the surface of the cell. And then down here, we have what are known as the apicomplexans. And the apicomplexans are the only one of the 4 groups of protozoa that are non-motile. They do not possess the ability to move. So I do want to point out some of the key protozoans that we will talk about this semester. First of all, we mentioned Giardia, and Giardia was the one that is detected, that flagellated protozoan that's detected in the patient fecal matter. We also talk about Plasmodium. Plasmodium is the cause of the disease malaria, and this is one of the greatest causes in history of human death worldwide from malaria. We also talk about toxoplasma or toxoplasmosis, which is that disease that you hear is acquired from cat litter boxes during the term of a female's pregnancy. So I want to get in and mention a few other pathogenic flagellates. We have the trypanosomas, and there's 2 different diseases. We have Trypanosoma brucei and Trypanosoma cruzi, which result in African sleeping sickness and Chagas disease, respectively. Entamoeba histolytica, which we'll get into much deeper this semester, causes dysentery. And we're going to end our discussion today getting in and looking at the helminths. And as we mentioned, the helminths are the most complex of the eukaryotes we will discuss this semester. They are multicellular and actually have some primitive systems, such as a digestive system and a nervous system. And these are strict parasites. They get into the host tissues, they attach with their mouth parts, and they actually reside and feed off the tissues while reproducing. And most of them are able to produce eggs and sperm. Some are hermaphrodites, meaning that they are possessing both sex cells on the same animal. The eggs go through a very simple phase. Once they're fertilized, they go into a larval period or a period of development before they enter the adult form and are able to pass on. We've got 3 major groups of helminths that fit into 2 different categories. We have the flatworms, which have several characteristics. They're flat, obviously, that you would have gotten from the name. They do not have a body cavity. Their digestive tract is a very simple pouch, and they have very simple nervous systems. And the flatworms are broken down into 2 groups. We have the cestodes, or tapeworms, which have these long ribbon-like bodies. And we have the trematodes, or flukes. The roundworms, on the other hand, are in their own group, and they look literally like an earthworm. These are the nematodes, and they have a very complete digestive tract and are able to hook with things like spines and hooks on their mouth, but their nervous systems are much more poorly developed than we see with the flatworms. Helminths are classified by many, many different mechanisms. Their shape, their size, whether they have particular organs or not, the presence of the hooks or suckers on the mouth part for attachment, how they reproduce, and their appearance of their eggs and larvae. So here's our helminths, and we have our tapeworms here, which we mentioned have this long ribbon-like body. Each of these little body segments is referred to as a proglottid. And the proglottids actually contain sex cells, such as the eggs. On the anterior end of the worm, we find the scolex, or the head region, that contains those hooks and suckers to attach onto body parts. We also have our other types of worms, which, again, very similar, have the oral mouth part up here, and have very, very simplified systems within their bodies. There's only about 50 species worldwide that are parasitic, and they are distributed all around the globe. They're acquired from either ingesting food or water, or they can be transmitted through contaminated soil as well. And they afflict billions of humans. If you are familiar with the show The Monsters Inside Me, which used to be on TLC, a lot of times they would feature organisms that are in that helminthic category. We talk a lot about the pinworm. So the pinworm is an example of a roundworm. The female worm is actually much larger than the male worm, and these actually grow and reside near the opening of the anus. And they emerge from the anal opening at night and actually cause itching to occur. And basically, you are self-infecting because as you scratch, you create fissures in the surrounding tissue that cause the worm to be able to spread to the surrounding areas. So with that said, I do also want to mention, as we talk about parasitic worms have something known as the definitive host. And the definitive host is the life cycle stage where the worm is considered to be sexually mature. So that's really important, and that's something we will talk about more this semester, is that definitive host, that sexually mature life cycle stage. So with that being said, that's the end of our presentation for Chapter 5. Feel free, as you're watching the video, to jot down some notes to bring to office hours, and I will address the questions at that point. Hope all is well, and I will see you all soon.